Welcome to this tutorial that begins Unit 6 of the course on complex brain functions, and we begin with a consideration of the associational cortex. I'd like to give you an overview of the associational cortex and tell you just a little bit about what features differentiate this portion of the cerebral mantle from the primary sensory and motor regions that we've studied heretofore. And in subsequent tutorials, we'll explore lobe by lobe what are the major features and functions of the associational cortices that are localized to each cerebral lobe. So, let's begin. And I want to first relate what we will be talking about today to our core concepts in the field of neuroscience. As we enter this final unit of the course, we are considering some of the most complex brain functions, indeed, those functions that we associate with human intelligence. And as this core concept suggests, our notions of intelligence can be broken down into different components. And it will be quite interesting to see over the next few decades how these various components relate to particular systems and circuits and functions of the human brain. But for now, we can state that human intelligence, and indeed intelligence in non-humans as well, arises as brains reason, plan, and solve problems. Well, we wouldn't have made it this deep into medical neuroscience if our human brains haven't endowed us with a natural curiosity to understand how the world works, including the world within your own brain. Well, congratulations to making it to Unit 6, and I hope that you find this to be a rewarding and satisfying unit where we will attempt to put a bit of a capstone on the learning that we've done throughout the course. And as we turn now to the associational cortex, in broad overview, my learning objectives for you are to be able to characterize the behaviors and the corresponding neural processes that contribute to cognition. And secondly, I want you to be able to discuss the major similarities and the major differences in the organization of what we will call primary cortex, that is primary sensory, primary motor cortex, and the associational cortex. Well, I think we're ready now to begin to talk about human cognition. And as we do, we realize that not everyone is on the same page with what we think we mean when we think of this term cognition. So I'd like to suggest first just a layperson's definition. I think we might all get on board with saying that cognition refers to the process of knowing. Well, in medical neuroscience, we want to try to express these ideas in neurobiological terms to the pretty stringent limit that we're able to do so given our relatively recent and quite imperfect knowledge of these underlying neurobiological mechanisms. Nevertheless, I do want to suggest more of a neurobiological definition of cognition. What I would suggest is that what we are speaking of are the neural processes by which the brain integrates meaningful stimuli, internal motivations, referring to brain and body states, and the product of this integration is awareness or perception, and, of course, the motivation of an appropriate behavior. So these are some of the mechanisms that operate in an instant as our brain is constantly processing the information that it receives in a context. That context reflects not just your individual experience, but also the evolutionary experience of our species, and indeed our phylogenetic lineage. Well, these are big concepts and big ideas. So I hope you will give me a little bit extra latitude as we pursue these ideas throughout this unit, knowing that much of what we talk about is really only an imperfect first approximation at the much deeper knowledge that undoubtedly will continue to accelerate over the next few decades. So if we're still both around and interested in medical neuroscience 20 years from now, perhaps we will have a much more precise and specific conversation. But for now, I think what we can do is begin to point towards what might be some of those key neurobiological mechanisms that underlie human cognition. Well, let's try to break this down just a little bit more. So we're talking about integrating meaningful stimuli. Well, these stimuli may be external, and they then might be detected by our special sensory systems. They may be stimuli that are internal and related to what we might consider to be the state of our brain and body. These states may relate to this internal schema of our body, which is itself a neural construct of the associational cortex. Well, to carry on, these brain and body states will reflect our emotions, our mood, our motivations, our memory, our repetitive drives, and even our injuries and disabilities. And I think that's an important concept for those of you that are in the health professions or pursuing the health professions to consider. That is, how might the state of body and brain be altered as a consequence of injury and disability? Sometimes I think it's very helpful as we understand the relation of body and brain to understand how do we close the loop on the experiences of the body as they feed back and have opportunity to shape or reshape the structure and function of circuits within the central nervous system. Well, we're talking about integrating these external stimuli with internal body states and producing awareness. And this awareness may be veridical, meaning that it might actually correspond to the physical reality that can be assessed and verified through other means. But more commonly, our awareness is highly subjective. Some would even argue that our awareness is influenced as much by our past experience or even our evolutionary history as it is by the presence of a particular stimulus that might be giving rise to energy that interacts with our special sensory systems. The integration of stimuli with internal states, the production of awareness, may ultimately lead to a response, to a change in behavior. And that behavioral response may result in somatic or visceral motor output. It may result in altered patterns of neural activity that represent knowledge or emotions, even without the overt expression of movement. So our brain is capable of representing the action, even if the action itself is not executed. Recall our discussion of the mirror motor system back in Unit 4. And as we will get to as we proceed on in Unit 6, we'll talk about vicarious representations of body and brain state that might be influential in our rational decision-making faculty. Okay, well, I go through all of this to give you a bit of a feel for what are the dimensions of cognition that we want to try to grapple with as we consider the organization and function of the associational cortex. Well, let me try to break it down for you even further. And if you've got your tutorial notes with you, you'll note that this is the table that I've provided for you. And this is really only a provisional framework for our discussions today. This certainly could be made much more specific and much more informative. But I think just to help us all get on the same footing, it's worth recognizing several different components to human cognition. And I'll suggest 5, beginning with the phenomenon of attention. So attention we can think of as a neural or behavioral function, and sometimes it's useful to give a metaphor or an example of what we mean. And I would suggest, as many others have before me, that one way to consider attention is to think of it as a searchlight or a spotlight, with the idea being that the neural mechanisms of attention help to focus or to filter this incoming stream of sensory signals so that we can zoom in or attend to only the subset of information that is most appropriate for driving behavior. Now, there are a variety of neural mechanisms that we now understand are relevant for understanding attention. Surely the networks of cortical neurons in the associational cortex of the parietal and frontal lobes in particular are relevant, but so are a variety of long projection systems that arise in the brainstem and in the base of the forebrain. And specifically what I have in mind here are the modulatory influences of brainstem reticular formation, thinking specifically about many of our biogenic amine neurotransmitter systems. Well, also relevant will be projections from the lateral portions of the hypothalamus that likewise can modulate brain state, or at least modulate the way neurons are processing information derived from other sources. The same general functions can be attributable to some of our long projecting neurons from the basal region of the forebrain, specifically the cholinergic neurons that project widely throughout the cerebral cortex and can influence the way cortical neurons respond to incoming sensory signals. So attention is just one important component of cognition. Another is recognition. And so by recognition, what I might imagine is being out in a crowd, perhaps in the marketplace or the town square or in a busy retail district, and you suddenly recognize the face of a friend in the crowd. Well, that moment of recognition is the result of a sensory processing stream. I suppose I have in mind here visual recognition, so we imagine that signals were processed in early visual cortex and increasingly higher stages of visual processing were engaged until in the ventral or inferior aspect of the temporal lobe, now the ability to unite the elements of the visual stimuli that comprise the human face are brought together, and those visual forms and features are bound up and recognized as belonging to an individual person. This is a neural process that involves the coding of feature representations in the primary cortices, but as I've just described for you, all the way through our higher order sensory cortices. And in this case, through the inferior part of the temporal lobe, that so-called WUT, or recognition processing stream, that runs through the inferior surface of the temporal lobe. So following the recognition of the face in the crowd, now you actually are integrating knowledge about this person that you are now recognizing. So we're bringing back memory together with the present experience of this stimulus in a process that results in actually knowing that friend. Not just recognizing the face, but now recalling information that's germane to understanding your relationship to this particular visual stimulus. And so this now is getting at the heart of what's going on in our associational cortex. It involves the association of disparate processing streams. So we've been talking in broad terms about the associational cortex. You'll see it in just a moment. Really, that word should give you much of what you need to understand about these territories of the cerebral cortex. They are associating inputs from a variety of different sources. And it's in that association that the integration occurs. And ultimately for us, this process of knowing can happen. Well, now that you've paid some attention, you've been able to recognize a face in the crowd, you know something about that person, now you're moved to action. So now I would suggest there's a planning phase that is a dimension of human cognition. And this planning, in this particular example, might lead you to actually approach and seek out that friend. So this is going to require executive decisions that are made principally in the associational regions of the prefrontal cortex. We'll talk more about that a few tutorials from now. But essentially, decisions are being made that will motivate your actions. So as the planning unfolds, so then follows the selection and the execution of the behavior. In this case, this may involve the actual approach behavior. that allows you to engage that friend in conversation. So this involves implementing both the short-term plan, for example, deciding how are you going to navigate through the crowd to meet up with your friend, as well as the long-term plan, or perhaps you're thinking about, oh, I'd love to talk to my friend about such and such a person or such a place. So there is a sense of both short-term and what might follow that begins to unfold. And this, again, is derived largely from the activities of our associational networks in the prefrontal cortex, now drawing out information that may be stored in memory in the other associational cortices throughout the cerebral mantle. Well, I hope this walkthrough gives you just a bit of a better feel for what we're talking about when we speak about human cognition, and it will provide you a bit of a context that we can recall as we begin to dive a little bit more deeply into the associational cortices of each of the four lobes. But before we get there, I just want us to now reflect a little bit on the anatomical organization of the associational cortex, and then that will be our point of departure into each lobe one at a time. Well, let's consider first of all really what we're talking about. And in case you haven't thought about it in these terms, when we've considered the organization of the human brain, especially the cerebral hemispheres, we have to admit that even though much of the course has now passed us, we've really only talked about 25% of the cerebral mantle. And that's about how much of the cortex is allocated to our primary sensory and motor regions, essentially those areas that are highlighted in yellow here in this figure. The rest of the cerebral cortex is what we call associational cortex. So all the regions of the brain that we see in our slide that are highlighted in blue, these are brain regions that are doing the job of associating. That is, they are involved in bringing together, integrating information that's being processed through lower to higher order divisions of our sensory and motor regions. Okay, one other point of review. I want us to recall the organization of the cortical microcircuit. As you will remember, undoubtedly, I suggested that the cortical microcircuit, this cortical columnar circuit that we find replicated throughout the entire cerebral mantle, performs three principal functions. It's involved in amplifying the input, which for the regions we've talked about thus far largely is derived from different divisions of the thalamus. There's also a computational function that reflects the operations of circuits mainly in the upper layers of the cortex. And then there is a communication function as information is conveyed from one cortical column to another, or from one cortical area to another, perhaps even across the corpus callosum or the anterior commissure, interconnecting a region of one cerebral lobe with its counterpart in the other hemisphere. Well, the cortical column, as you will recall, is made up of a large number of neurons. There are some neurons, such as the stellate neurons and these pyramidal neurons, which are excitatory. They release glutamate as a neurotransmitter. It binds to kinds of glutamate receptors that we've been talking about throughout the course, such as our AMPA and our NMDA receptors. Not shown here in this illustration are small interneurons that typically release GABA, gamma-aminobutyric acid, as a neurotransmitter. Those would be inhibitory interneurons in the adult brain, where the chloride concentrations allow for the stabilization of membrane potential below threshold, which is the definition of an inhibitory postsynaptic potential. So these pyramidal neurons are the ones that project for considerable distance across cortical columns, from one cortical area to the other, even from one region in one hemisphere to the corresponding region in the other hemisphere. Or in the case of our motor system, from the motor cortex down through the brainstem and even into the spinal cord. So it's the excitatory neurons that give rise to the longest projections within the central nervous system. The smaller inhibitory interneurons tend to give rise just to local connections within a cortical column, or perhaps across a limited number of cortical columns in the nearby region within a single cortical area. Now, one aspect of this cortical microcircuit that I've not yet really talked about very much is the fact that while for many regions of the cerebral cortex, their principal input is derived from the thalamus, all regions of the cerebral cortex are also receiving input from other parts of the cerebral cortex. And for those cortical areas that are at a higher level of processing, those cortical inputs are being sent into the middle layers of the cortex, right along with the thalamic input. Well, this now becomes an important distinction between the organization of, let's say, a primary sensory cortex and the associational cortex. For the primary sensory cortex, the predominant source of input is from the thalamus. So when we talk about amplification, we are mainly talking about amplification of input signals derived from the relevant thalamic nucleus. Well, when it comes to the associational cortex, we have to recognize that an additional major source of input will be other cortical areas. Now, for many of our cortical areas, the bias in input is significantly shifted in favor of these other divisions of the cerebral cortex. There remains a thalamic component to inputs for each location in the cerebral mantle. So then, for the associational cortex, it's other cortical regions that are providing the input that provides the basis for the amplification that is happening within the cortical column. Well, it's also this input from other regions of the cerebral cortex that is being computed and from which new cortical properties are arising as integration and association is happening within these cortical networks. And then lastly, the communication piece likewise pertains to the operations of circuits that are giving rise to long-distance projections across columns, across areas, perhaps even across the hemispheres. So I'd like to show you just an overview slide that helps to put the anatomy of the associational cortex in context. So our associational cortex, as I mentioned, does receive inputs from the thalamus, specifically a part of the thalamus that we call the mediodorsal thalamic nucleus, as well as posterior structures, including the lateral posterior nucleus and the pulvinar. But these thalamic nuclei are a bit different than those that we've described in our sensory and our motor pathways. You may remember when we first introduced the thalamus some time ago at the start of Unit 3, we talked about first order and then higher order thalamic nuclei. Well, the first order thalamic nuclei would be those that provide input into our sensory cortex. The higher order thalamic nuclei are those that we now have in view when we think of the associational cortex. So these would be thalamic nuclei which themselves are driven principally by inputs from the cortex. So these thalamic nuclei, while providing input into our associational cortex, seem to be consistent with the relay of information from lower order areas to higher order areas. Only in this case, the relay involves a loop from cortex to thalamus, now back up to higher order or associational cortex. So if we return to our overview image, now we can understand perhaps a bit better how this thalamic input fits into the story. So the thalamus is providing input to the associational cortex, but we imagine that it is consistent with input from other divisions of the cerebral cortex. And it's these other divisions that are principally driving the computations that are being performed in these associational networks. So these associational networks have interactions with regions in the opposite hemisphere, some of which are homotypic, that is, they are the comparable anatomical unit found in the opposite cerebral hemisphere, but the projections often are a bit more widespread. They may involve non-homotypic or non-corresponding areas. The subcortical connections of the associational cortex are similar to what we've been describing, mainly focusing on our sensory and our motor systems. That is, the associational cortex provides input to the striatum of the basal ganglia. Not so much the putamen, but as you may remember, we briefly mentioned the caudate nucleus and the nucleus accumbens as divisions of the striatum that were involved in motivating or moving our thoughts, our cognitive faculties, our emotions. Those are the divisions of striatum that we principally have in mind here. Likewise, there are inputs that are relayed into the cerebellum from the associational cortex, undoubtedly involving our lateral cerebellar hemispheres. This is a fairly newly discovered aspect of cerebellar function that perhaps pertains to cognition and emotion. Still a somewhat controversial area, but one that the anatomy would seem to bear out. So one last source of input to the associational cortex that I would highlight here are the inputs that are derived from our brainstem modulatory systems. And so what I have in mind here would be, for example, the noradrenergic neurons of the locus ceruleus, the serotonergic neurons of the raphe nuclei, and especially now for the prefrontal cortex, the dopaminergic cells of the ventral tegmental area. All of these brainstem systems have widespread projections to the cortex, and they can provide an important modulatory signal that might shape the way the circuitry of the associational cortex responds to the principal inputs that are derived from other cortical regions. In addition, there is input from these cholinergic cells that are found in a particular region of the basal forebrain that we call the nucleus basalis of Meinert. So these are neurons that seem to be important in maintaining attention and driving other aspects of cognition. This is the neurotransmitter system that is the target of the pharmaceutical interventions that are presently approved, at least in the United States, for treatment of mild cognitive impairment and Alzheimer's disease, specifically the cholinergic system that is degenerating in this devastating neurological condition. One interesting approach would be to try to increase the levels of acetylcholine, even though the neurons that are producing and releasing acetylcholine are being lost to disease progression. So the interventions that are available are an attempt to either activate the receptors for acetylcholine, the nicotinic acetylcholine receptors that are present in the brain, or to sustain what levels of acetylcholine may be present by inhibiting the enzymes that break down this neurotransmitter within the brain tissue itself. Well, these interventions unfortunately don't halt the progression of the disease, but they may prove beneficial in supporting cognition, especially in the early stages of the disease when quality of life can easily go in a downward direction without the proper support that might come from multiple sources. Well, I hope this overview has given you a sense of what our discussions will be about here in Unit 6 as we begin to think about the brain basis of cognition. And I hope it will give you some orientation to the tutorials that will follow, where we will try to explore systematically the associational cortices of the parietal, temporal, and frontal lobes, and then eventually try to tie this together as we think about the way that brain state can be modulated and how brain state can be integrated in the production of human emotion. So I look forward to working through Unit 6 with you, and look forward to what discussion we may have on our website. So I'll see you next time when we will consider the associational cortex in the parietal lobe. Welcome back to Unit 6 of Medical Neuroscience on complex brain functions. And in this tutorial, I'd like to talk to you a bit about the associational cortex that we find in the parietal lobe of the human brain. Our tutorial relates to two of our core concepts in neuroscience. We are talking about the associational cortex, and it's from the integrated action of these networks in the brain that intelligence arises as the brain reasons, plans, and solves problems. And as I said last time, we wouldn't be here, and you certainly wouldn't be with me this deep in medical neuroscience, had it not been for the innate curiosity that your brain has given you, which gives you a desire to understand how the world works, even the inner workings of the human brain itself. So I appreciate your efforts, and I certainly am right there with you in celebrating the curiosity that our brain has endowed us with. Okay, well, moving into our learning objective for today, there's just one objective, and that is for you to be able to discuss the major functions that are localized to the associational cortex of the parietal lobe. Well, I'll remind you that when we are speaking of the associational cortex, what we are referring to really is the 75% or so of the cerebral mantle in each hemisphere that is beyond our primary sensory and motor region. So for our purposes here, we are focusing in on the parietal lobe, which is posterior to the central sulcus, superior to the lateral fissure, and then bounded from the occipital lobe by this imaginary boundary that we recognize as the parieto-occipital sulcus emerges from the dorsal midline and extends down to this feature that we call the pre-occipital notch. And the pre-occipital notch is formed by the tentorium as it impinges upon the developing ventral surface of the hemisphere. Well, so this is our parietal lobe, and specifically we're concerned with what we might call the posterior parietal lobe, which is that territory that lies posterior to our primary somatic sensory region, anatomically speaking, posterior to at least the crest of the postcentral gyrus, if not posterior to the postcentral sulcus. So if I can just show you an image of the human brain using my model here. Here again is our central sulcus. So this would be our postcentral gyrus. So we're talking about the formations of the posterior parietal lobe. And if you remember back from Unit 1, the fingers on the gyri experience, I suggested that you can conceptualize the parietal lobe as the cortex that's posterior to the central sulcus. So the space between my first and middle fingers is the central sulcus. My index finger would be the postcentral gyrus. So what we're talking about today basically is the cortex represented by my index and middle fingers of my left hand. The index finger is representing the superior parietal lobule, and the middle finger is representing the inferior parietal lobule, with the sulcus between these lobules being the intraparietal sulcus. So if you don't recall that aspect of human brain anatomy, why don't you spend a few minutes back in Unit 1 and refresh your memory of these structures. Better yet, maybe you can find a large piece of paper and a whiteboard and once again draw the lateral view of the human brain to make sure you're on board with these divisions of the parietal lobe that will be our focus in this session. Okay, well, that is the focus for today from an anatomical point of view. Now let's talk about function. So when we consider the functions of the posterior parietal cortex, the principal function that we want to think about is attention. So recall from our brief survey of various components of cognition, we started with attention, this cognitive searchlight, this ability to illuminate a limited region of our world and to focus on the information derived from that region. Now, when we speak specifically of the role of the parietal cortex in attention, I want you to understand that I'm not claiming that all of attention is seated in the parietal cortex. That's not the case at all. Attention is a complex and multifaceted neurobiological mechanism that is broadly distributed throughout the brain. Rather, the role of the parietal network seems to be to direct our attention to particular locations in the outside world, or perhaps to the body surfaces as it interacts with that world. So it's really the direction of attention that is principally the function of the posterior parietal cortex and the associational networks of this lobe. So let me give you an example of the role of the posterior parietal cortex in directing our attention. I'll show you some data that is illustrated in our textbook. So this is figure 26.9, and it illustrates how attentional mechanisms in the posterior parietal cortex can increase the firing rates of neurons in response to stimuli that happen to be within that cognitive searchlight. That is, that happen to be the object of the subject's attention. So this is an experiment done in a non-human primate model, and a microelectrode is inserted into the posterior parietal cortex, and action potentials are being recorded. And what we find in panel A are post-stimulus time histograms reflecting the activity of these neurons when the monkey is instructed to either attend to the location of the stimulus or to not attend to the location of the stimulus. So as we can see very clearly and definitively, when the animal is instructed to ignore the target stimulus that is otherwise present within the receptive field, you'll notice that there's only a modest increase in activity compared to what happens when the animal is given the cue to attend to the target. Now there's a huge increase in activity for that location in the visual world that has captured this animal's attention. So what we see by various behavioral measures is that this animal is directing attention to a particular stimulus, and that attention can be measured in terms of the amount of time that the animal is dwelling on the target, as well as the amount of reward that the animal receives for doing the behavioral challenge that the experimenters have put forward. Now, when we relate these behavioral metrics to neuronal firing, what we discover is that neuronal firing is rather tightly related to the behavior as indicated by the amount of reward that the animal has received. And this simply indicates that attention can have an effect on the firing rates of neurons that are responding to sensory stimuli. And what attention does is shifts, in effect, the dose-response relationship or the gain of that function. So with no particular direction of attention or anti-attention, that is, directing the animal to ignore the stimulus, we imagine that this function would be much more flat. But what attention does is it increases the gain of this relationship between stimulus and neuronal response, or in this case, between the location of the stimulus and the behavior that's motivated by that stimulus. So, I sometimes reflect upon this and my own behavior, and as an educator, I get to meet lots of interesting young people, and unfortunately, I don't do very well remembering their names and associating their names with their faces. And, well, sometimes we say, I'm just terrible with names, and don't hold this against me. But really, I look at these data, and I think, maybe I'm just not paying attention. And that's something I can do better. So I want to commit myself to paying more careful attention, to being, as my communications colleagues like to say, to be in the moment, where our attention is focused on the task at hand, be it a sensory task or a cognitive task, an emotional task, whatever it happens to be. I would encourage us all to be in the moment and to reflect upon how attention can change the gain of our response to a given set of stimuli. Well, the same basic effect can be seen within our early sensory cortex, and this is an example of how attention can modulate the tuning properties of neurons even in the early visual cortex. So what we're looking at here is a tuning function. So we call this a tuning curve, in this case for orientation of the visual stimulus. As you remember, neurons are selective for particular orientations of visual stimuli in particular directions of stimulus motion. Here we're looking at the tuning for the orientation of a stimulus. And this neuron is tuned somewhere for probably a visual stimulus that is more or less vertical, but perhaps tilted in the leftward oblique direction. Now notice what happens when the animal is given a cue to pay attention to the orientation of a particular location of the visual world. What we see is for those neurons whose receptive fields are being stimulated, the tuning function gets much sharper. So this neuron is becoming more selective and more responsive as a function of this attentional signal. Now, what we don't really understand in full yet is, well, where does this attentional signal come from? What's responsible for this effect where we see an increase in the response and selectivity of neurons, even in early sensory cortex? Well, we imagine that this parietal associational network has a significant role to play. Perhaps there are feedback projections from parietal cortex into occipital cortex. Perhaps there are systems that allow the parietal network to recruit other modulators of attention, such as those basal forebrain cholinergic neurons that we spoke about last time, or our biogenic amine systems in the brainstem. We don't quite yet know how all this works together, but we at least have a sense of who the major players are in mediating these attentional effects in sensory processing. Well, one dimension of attentional processing in the parietal lobe that has garnered quite a bit of interest by clinicians, but also by cognitive neuroscientists, refers to an asymmetry of attention that is often revealed in patients that have damage to the parietal associational network, particularly when that damage is localized to the right hemisphere. Now, I really hesitate to do this because for a variety of reasons I'm uncomfortable representing a patient or pretending to be a patient with impairments that I don't actually have. I think that's a complex business and I don't do it lightly. But I do want to illustrate for you a couple of simple clinical tests that are used to assess the possibility of there being an attentional problem with patients with parietal lobe injury. So here's a very simple test, and this test simply is to bisect the middle of the line. So presumably you and I would survey the challenge and just draw a line somewhere around there. Well, this is what a normal person would do. But an individual that has a condition that we call hemi-neglect syndrome might be inclined to put their line somewhere over here. So there's a significant rightward shift in the perceived assessment of the center of this line. Well, here's another challenge, a little bit more demanding, which you may find yourself doing with some patients, depending upon their capacity to move and articulate their drawing hand. But you might ask this patient to simply copy a scene. So here's a photograph of our fabulous Duke Chapel that we have here on campus that I took not long ago. And I may be asked to copy this scene, and if I were such a patient that had parietal lobe injury, I may look at this and I may say, okay, well, my attention is certainly drawn to the chapel tower, and I may begin to do something like this. I may look a little bit more detail. There's some shadow in here. Well, there's some trees in the foreground, so I'll try to represent that somehow. And I'll, maybe continue to add some detail over here. And as my visual gaze, my visual search is changed, I might eventually come over here. I might add a few of these spire details. I might return and draw some more shadows in here. And I think you basically get the idea that what I am inclined to do is to illustrate features that are largely present over on the right-hand side of this scene, without providing much detail, if at all, for what might be left of the midline of the actual visual scene. Okay, here's one additional test that often is quite striking and sometimes, frankly, somewhat disturbing when this is observed. You've probably noticed that I have a clock on my wall, and you've heard the ticking over our time together. There's my clock I hope you can see the blue devil on the face of the clock. Well, one can be asked by a clinician in a simple test to simply draw the clock face. So obviously this can be done by a person with an intact nervous system. They will draw a circle and they will begin to think about, okay, well, what does a clock look like? Well, the number 12, at least in our Western cultures, is up above, 6 is to the bottom, 3 is to the right, 9 is to the left, and so on. And someone might be asked to draw a time like 9 o'clock, and you'd say, okay, well, the large hand is pointing towards 12, and the short hand is pointing towards 9. There's 9 o'clock. Well, if one were to ask a patient to do the same challenge that has had damage to their parietal associational network, especially on the right hemisphere, here's what one might draw. One might know perfectly well that the clock face is round. There are numbers 1 through 12. So number 12 is on top. Now, what the patient very well might do is to place all of the numbers from 1 to 12 on the right-hand side of the clock face. And if asked to demonstrate 9 o'clock, the knowledge of the position of the hands might direct the patient to make a drawing that is clearly incorrect in some way, but also clearly correct in terms of the rules about where the large hand and the small hand of the clock would be pointing. Well, these are just some simple representations of what a patient that has had damage to the right parietal lobe might present on simple clinical evaluation. Of course, there are many more sophisticated means of doing diagnostic neuropsychological testing. That's beyond the scope of my presentation here. Rather, I want to emphasize to you simply the functional localization of damage that produces this so-called contralateral hemineglect syndrome, often called left hemineglect syndrome because it's most frequently the left side of body and world that is the object of neglect. Well, here's a simple compilation of a series of patients that have presented with hemineglect syndrome, and as we can see, there seems to be a region of overlap that is focused here in the inferior parietal lobule of the right hemisphere. That's not to say that damage of this region is necessary to produce a hemineglect syndrome. Indeed, one might have damage to the frontal cortex, perhaps damage into the temporal cortex that can be associated with hemineglect syndrome. But the most common network that is impacted with brain injury that results in the hemineglect syndrome seems to be focused right here in this inferior parietal lobule of the right hemisphere. And for those of you that recall the actual gyri of this region, we're talking about an impact on the supramarginal gyrus and just behind it the angular gyrus. Well, why should this be the case? Well, we think this is the case because in all of us there is very likely an asymmetry to the attentional functions of our parietal associational networks. And that asymmetry is illustrated with these ray lines that are shown in this illustration from our textbook. Notice that for the left parietal cortex, attention is focused across the midline to the right side of the visual world. However, for the right parietal cortex, attention is distributed throughout the entire visual field. There may be a bias towards the contralateral side, that is, attention may be focused more particularly on the left side of the visual world than the right, but there does seem to be an asymmetry. This can be revealed in functional brain imaging studies where subjects are directed to attend to one side of the midline or the other. And what we discover is that when subjects are directed to attend to the left side of the visual field, the right parietal cortex increases its functional activity. However, when subjects are directed to attend to the right side of the visual field, what we find is much more bilateral activation of the parietal associational network. Well, there are consequences of this asymmetry for patients that have damage. For example, imagine a patient that has had damage throughout the right hemisphere, including, of course, that inferior parietal lobule of the right side. One might expect a severe neglect of the left side of the visual world because we've now taken out the capacity of that parietal network to direct attention across the midline. One might still have a capacity to attend to the right side of the visual world because of preservation of the left parietal network and its ability to direct attention to the right side of the visual scene. Now, it's much more common to find left hemineglect than it is right hemineglect. And again, this is reflecting the fact that with damage to the left parietal associational network, there's still the capacity to direct attention to the right side of the visual world. And that again is related to the fact that this right parietal cortex also can direct our attention to the ipsilateral field, that would be the right field, in addition to its capacity to direct our attention to the contralateral or to the left visual field. Now, I've been emphasizing vision and visual field, but I want to make the point that hemineglect is not just about vision. It's not just about seeing the world on the outside. Patients with contralateral hemineglect may also fail to appreciate the significance of their own body parts that are on the left side of the midline. Imagine the challenge of rehabilitating a patient that perhaps has had a stroke involving the right middle cerebral artery that may have affected sensory motor control of the left arm and hand. So this patient is in need of neurorehabilitation. But should there be a hemineglect syndrome due to damage of the nearby right parietal associational cortex, this patient might actually deny impairment of that left arm. Furthermore, the patient may not even recognize that left arm as part of themselves. You might even find the disturbing situation of a patient who gets up in the morning and only dresses the right side of their body. It's hard to imagine what might be going on in the networks of a person with such an injury. It's not that they have a visual deficit. It's that they fail to appreciate that what is here actually belongs to them. There are certainly stories in the medical literature of patients who have assumed that this body part belongs to someone who must be behind me and sticking their left hand in my visual field. Well, I think what this does is it goes to illustrate that the concept of self, the brain's construct of the body's image, is in fact the product of associational networks that are largely localized to the parietal cortex, but of course also extend throughout each of the lobes of the human brain, including the networks that are localized to the insular cortex, which are mainly concerned with our internal visceral state. Pardon me for bearing a little bit of my skin. I hope that wasn't offensive, although I suspect it may have been to some, but maybe that will help emphasize the importance of appreciating the associational functions of the parietal cortex with respect to attention. Well, the last point that I'd like to emphasize with you before we conclude is that indeed the parietal cortex is critical for building this body image, this body schema, this neural construct of self. You know, perhaps one fact of our existence that we most often take for granted is that our brain is embodied. We are embodied cognate creatures. That is to say that our sense of self is intimately related to this body that we use to interact with the world around us. In that sense, is subject to injury and disorder with damage to the posterior parietal networks. And this can lead to some rather bizarre and intriguing phenomenon, as we've already discussed some, with respect to phantom sensations when there is nerve injury or limb loss. And now we see the hemineglect syndrome, which reflects also a disordered body schema. And this can present in very extreme forms that some of you may want to go on and read about, including people that seek surgical amputation of body parts. Evidently, because in their evaluation, they would be more complete as a person, as self, without that body part attached. Very likely, these are people that have dysfunction and disorder, perhaps disease, afflicting the associational networks that produce this sense of self embodied in the networks of our posterior parietal cortex. So I'll conclude this tutorial now, and when we resume, we'll be looking into the associational networks of the temporal lobe. So thanks for listening, and I'll see you next time. Welcome back to Medical Neuroscience and Unit 6, Complex Brain Functions. In this tutorial, I'd like to explore together with you the associational cortex of the temporal lobe. This topic pertains to two of our core concepts in the field of neuroscience. We are exploring the associational cortex, and it's from these vast networks of cortical regions in the human brain that intelligence arises. These are the parts of the brain that are in significant ways responsible for our capacity to reason, to plan for the future, and to solve problems. This is also the networks of brain regions that gives us the capacity for curiosity. And this is what has driven us all together as a learning community, and for that I'm very grateful. Well, my learning objectives for us in this tutorial are to discuss the major functions that are localized to the associational cortex of the temporal lobe. I want us to be able to focus on a few of those functions in particular. One of them will be human memory. So I want us to be able to differentiate categories of human memory and discuss the relevant neuroanatomical systems that support these different forms of memory. I want us to focus a bit in this tutorial on language and how language function is represented in the brain. And there are two very famous parts of the brain that we'll discuss today, Broca's area and a region that we call Wernicke's area. And I want you to be able to differentiate the functions of these two different regions and how they contribute to language function, and also differentiate the problems that people who experience injury to these brain systems experience. So I want you to be able to differentiate the aphasias, that's the medicalese term that we use to describe language problems associated with one of these regions or the other. So objective 3 is to differentiate Broca's and Wernicke's aphasia. And then finally, our fourth learning objective in this session is to focus on the concept of dementia. Dementia is a syndrome that characterizes disordered cognition. So I'd like for us to think together a little bit about dementia and just very briefly discuss the most common cause of dementia. That would be Alzheimer's disease. So I want you to be able to relate the various signs of dementia to specific regions of the associational cortex that we're now learning about. Well, let's begin again, as we did in the last session, by just considering the distribution of cortical areas in the cerebral mantle. And the basic point to bear in mind is that as we describe the associational cortices, what we're really concerned with is the vast stretches of the cerebral cortex that are between the primary sensory and primary motor regions. So this really accounts for, again, about 75% of the entire cerebral mantle that is present in the two cerebral hemispheres. While much of Unit 3 of the course was focused on sensation and sensory processing, we really hadn't yet gotten into a full description of how, for example, the somatic sensory cortex feeds signals into the parietal associational cortex, and likewise, we've only briefly touched on how the visual cortex feeds signals into the parietal associational cortex as well as the temporal associational cortex. Well, today we're going to be talking about the temporal lobe. So we could easily spend entire sessions just on several of the important functions that are associated with this temporal cortex. So unfortunately, time and space does not allow, but I hope to give you at least a bit of a feel for some of the important aspects of cognition that are dependent upon the operation of networks that we find in this temporal associational cortex. And I hope to whet your appetite, and I hope that that appetite will be satiated in other learning experiences that you might pursue should your interest lead you in that direction. Okay, well, I think we're ready to begin our discussions of the temporal associational cortex, focusing on one particular domain of function at a time. And so what I'd like to turn our attention to now really is the associational cortex that we find on the inferior surface of the brain. And it's here that we have networks that are receiving input largely from the visual system, but also from the auditory system, that are responsible for building up our ability to recognize a sensory stimulus, be it a simple sensory stimulus such as the shapes that define an object in the visual field, or something more complicated like a human face. So let's turn our attention then to the inferior portion of the cerebral cortex that we find here on the temporal lobe. And it's there in the human brain that we find these regions that have been well studied in non-human primate models. And what I'm referring to here is this ventral object recognition pathway. You may recall that we spent just a little bit of time talking about this when we considered the central processing of visual signals in Unit 3. And what I described for you is the elaboration of a series of higher order visual processing centers as one progresses from early visual cortex, that would be our first primary visual cortex, Brodmann's area 17, Brodmann's area 18, our secondary visual cortex. And then from here, signals are sent down through a network of regions that are in the inferior part of the temporal lobe. Now, just so that we get the anatomy straight, in the non-human primate, these regions are associated with the inferior temporal gyrus. In the human brain, these regions tend to be more on the ventral aspect or the inferior aspect of the temporal lobe. The inferior temporal gyrus contributes to this network, yes, but in the human brain there are additional regions that I'd like to highlight for you, namely associated with the lateral occipital temporal gyrus. And if we look at my model of the human brain, what I'm really talking about is the region of the temporal lobe that we find on the ventral surface of the brain. We see just a little bit of the relevant region just peeking in the anterior side of the cerebellum. But to really talk about what we have in view here, I need to actually do a little dissection here of this model. So I can remove the cerebellar hemispheres and show you what I'm referring to. Actually, I'll just remove the one on this side and highlight the gyral formations that we find here on the inferior side of the temporal lobe. And specifically, there's a long gyrus that runs the extent of the inferior aspect of the temporal lobe down into the occipital lobe, called the occipital temporal gyrus, or sometimes the lateral occipital temporal gyrus. And this is where we find this ventral visual processing stream in the human brain. In fact, there's a region of the posterior part of the lateral occipital temporal gyrus that's sometimes called the fusiform gyrus because of its shape that's often seen in many human brains, a spindle-type shape or a fusiform shape. So that's where we find these regions in our brains, and when we look at an image of a non-human primate, sometimes we get perhaps the erroneous impression that the relevant areas are somehow out here on the lateral aspect of the temporal lobe. In our brain, that's where we would find networks that have more to do with understanding language than visual recognition. So I just wanted to be clear about that anatomy. In fact, if you'd like to have a look at the human brain again, I might encourage you to view our tutorial session from Unit 1, looking at the ventral side of the human brain. In fact, it would be awfully nice to have a quick look at that right about now. So, I wonder what the human brain actually looks like. So this bit of brain is the temporal lobe. And on its inferior surface, we have a long gyrus that extends throughout the length of the inferior temporal lobe back towards the occipital lobe. That's called the occipital temporal gyrus. Sometimes there's a small sulcus that divides it into a lateral and medial components. And then along the medial edge of the temporal lobe, there's a structure that we call the parahippocampal gyrus. And deep in the anterior part of the temporal lobe, in this position, this parahippocampal gyrus folds in upon itself into a structure that we call the hippocampus. And we'll see the hippocampus when we look at sections through the brain. Now, the occipital temporal gyrus and the parahippocampal gyrus extend down into the occipital lobe. And at some point in the occipital lobe, we define a region that is a boundary between the lingual gyrus near the midline and what remains here of the occipital temporal gyrus. Sometimes this region of the inferior occipital lobe is called the fusiform gyrus because of the superficial shape that is sometimes appreciated as these sulci come together. Well, that was a nice refresher. It's always pleasant to be confronted again with the actual form of real human brain specimens as we continue to improve in our mind this mental image of the structure of parts of the human brain that otherwise we might not explore in great detail in this course, such as this inferior aspect of the temporal lobe. Well, so let's talk a little bit about the actual recognition pathway itself that we find here. Well, in non-human primates, this has been very well studied using stimuli that we think would be meaningful to a non-human primate. But of course, what we'd really love to do is be able to characterize the function of these regions in our brains. And we've been able to do that with our brain imaging techniques such as functional magnetic resonance imaging. And when these experiments have been done, what is quite clear is that there are regions in this inferior temporal lobe, again associated with this so-called fusiform gyrus region, that seem to become especially active when we are confronted with a human face. So a human face is a wonderful stimulus for us as social creatures, and indeed for many non-human primates as well. The human face provides a complex visual structure that conveys great meaning, and that meaning is often best understood in a social context. However, typically an important step towards interpreting that social context requires the recognition of the identity of that face. We want to know, is this face friend? Is it family? Is it foe? And these recognition events are what lead us towards an understanding of the intentions of this person who's confronting us. So recognition is a key function of this inferior temporal network, especially when it comes to visual information, and especially for us when it comes to the human face. So these are examples of the kinds of specialized networks that we find in this inferior temporal lobe. There is a network that seems to be especially responsive to faces. There are others that might actually be specialized for the recognition of inanimate objects of various types. And there may be multiple categories of object recognition networks that we may find in this aspect of the human brain. This remains an active area investigation, and it's really a fascinating field to follow. So if you're interested in recognition and in human faces in particular, I would encourage you to get into this aspect of neuroscience, at least through study of the literature. And it's a rapidly evolving field, and as you might imagine, there's a lot of interest in understanding how it is that we come to recognize and interpret meaning in these complex stimuli, especially those associated with the face and facial expressions. Well, as we've been discussing throughout our sensory and motor systems, one can experience particular neurological signs and symptoms with damage to these cortical networks. And in this case, damage to these recognition pathways can lead to a phenomenon that we call agnosia. Agnosia is a Greek term that means not knowing, and it can be modified by a variety of prefixes that describe the specific modality of information that is missing from the cognitive awareness of the subject. And in the case of this region, that seems to be specialized for the recognition of human faces, damage to this inferior temporal cortex can lead to an inability to recognize a human face. This is called prosopagnosia, which, to expand upon the Greek there, and those of you that are native speakers, perhaps you can chime in with additional nuance of the fantastic detail and beauty of the Greek language. But for our purposes, we can define prosopagnosia as not being able to recognize the personhood that is represented by this stimulus, this sight of a human face. Well, let's get back to the non-human primate model, because I'd like to show you some data that helps us understand why prosopagnosia would occur with damage to this part of the brain. Some years ago now, dating back to the early 90s, neuroscientists began to record from neurons in this inferior part of the temporal lobe. And again, in the non-human primate, the relative regions would be in the inferior temporal cortex associated with this inferior temporal gyrus. And it was discovered that neurons could be found there that were responsive to visual stimuli, but not simple visual stimuli like moving lines and patterns and textures, but more complex stimuli that often conveyed some understanding of the world in which we interact with. And certainly for non-human primates, an important stimulus for these higher order associational areas is the sight of a face. And what was perhaps surprising is that neurons responded vigorously well to not only the presentation of a conspecific face, but also to the presentation of even a human face. So here are post-stimulus time histograms that show us the responses of a neuron to the presentation of facial stimuli, which is indicated by the presence of this green bar in the background here. So it's quite interesting to note that variations in the details of these faces, such as the face presented in Trial 1, Trial 4, and Trial 5, still elicit vigorous responses, even though some of the details have been changed. But when the elements of the image are changed such that we don't recognize the stimulus as presenting a cogent human or non-human primate face, then the response is severely diminished. That would be what we see here, for example, in trial number 2, where the elements of the face have been scrambled. Now, quite interestingly, in trial number 3, there's still a vigorous response in this neuron, perhaps not as much with a full view of the face, but it's consistent with our capacity to fill in the missing details and still achieve the job of recognition. So trial 3 actually makes a pretty good case for this neuron being interpreted as involved in recognition of this stimulus rather than in the details of the textures and the orientations and the movements of the elements that comprise that face. Those would be more lower order visual cortical functions. Here in the inferior temporal gyrus, and for us in that lateral occipital temporal gyrus, these neurons are recognizing the identity of this face. Now, of course, it's a bit more challenging to understand what this might mean for a non-human primate, but one can do additional variations of these experiments to try to understand more about coding of faces in this part of the brain. One experiment that has shed significant light on the way information is coded here has been to vary the view of the face from a full frontal view to a full profile view. And what was discovered is that there are indeed neurons that seem to be selective for particular views of the face. So, for example, in this figure from your book, we find recordings from a neuron that seems to prefer these profile views here in trials 4 and 5 compared to the full frontal view in trial number 1. So why would that be important? Why should you want to be able to recognize the face in profile versus the face in frontal view? Well, there may be quite different kinds of social cues conveyed via a frontal view of a face compared to a lateral or profile view. One can interpret intention through the frontal view in a way that is perhaps less clear in the profile view. On the other hand, understanding a profile view of a face can convey its own meaning. One might be interested to know, what is this individual looking at? Perhaps there's something that I might want to look at. So we continue to build up this model of intentionality based on the social cues that are associated with the face, be it a frontal view or a profile view. So I think it makes a certain amount of sense that we ought to have some selectivity in the ability to recognize not just the presence of a face, but the meaning that is presented in that face. Well, let's switch gears now and move on to a consideration of memory function. As we talk about memory very briefly here in this tutorial, I think it's important to be clear really what kind of memory are we talking about. And it's possible to differentiate different categories of memory. In fact, it's quite helpful to do so because, as it turns out, different forms of memory are supported by different networks in the brain. So one can imagine that there is a kind of memory that is available to conscious cognition. So what I have in mind here is the kind of memory for people, for places, for the episodes of our lives, for the things that can access our language faculty and allow us to describe verbally the content of that memory. And so this kind of memory we call declarative memory. It's available to conscious processing. It reflects the daily episodes of our lives, the semantic symbols that give meaning to our thoughts, and we can access those symbols and apply them in the form of language. So with declarative memory, we have access to our lexicon that allows us to report the content of that memory. It provides a personal history of our life events. So declarative memory, sometimes called episodic memory, is that memory for which we can declare its content. It reflects the daily episodes of our lives. Now, this is to be contrasted with another category of memory, really multiple subcategories of memory, that in our textbook we simply choose to call non-declarative memory, but it can be subdivided in various ways. Sometimes this is called implicit memory. Some subdivisions of this form of memory can be called motor memory, so the nomenclature is actually not entirely consistent across various sources. So for those of you that are in this field and prefer a particular nomenclature that might be different from this one, I hope you'll pardon my use of the nomenclature that we present in our book. But I think it's helpful to just make this broad point that it's important to differentiate from declarative memory these other forms of memory. So these are forms of memory that, generally speaking, are not available to consciousness. That is, it's much more difficult to access our lexicon, to put our semantic labels upon the content of this memory. So in this category would be motor skills, for example. Learning to ride a bicycle, learning to ice skate, or learning to do some other motor skill that once acquired seems to be with us perhaps for life. Of course, depending upon how often we practice and resurrect that skill. So it's quite challenging, is it not, to put into words the nature of the skill that allows us to ride a bicycle. We can describe riding a bicycle, but we cannot access the content of that skill through our semantic faculties. This is an important clue that this kind of memory might actually be acquired and consolidated through a different kind of brain system than is operating on our declarative memories. Well, in addition to motor skills, there are various associations that are established, either explicitly or implicitly, and it's those implicit associations that are exemplars of non-declarative memory. There are various phenomena related to priming that is a form of memory that is non-declarative. And related to motor skills, there are puzzle-solving skills that are accrued through experience and practice that, like other kinds of motor skills, are not readily accessible to our language faculties. Now, there's another dimension of memory that can be categorized, and that is the temporal dimension of memory. There's a sense of memory that's immediate, and this is memory for the previous few moments that are essential for maintaining consciousness, conscious awareness, conscious deliberations over cogent thought. So this is a form of memory that most likely reflects ongoing recurrent processing throughout cortical networks. But there's another form of memory that helps us to maintain the goal of our behavior. And so this is now extending over not just the last few moments, but perhaps seconds to minutes. This we call working memory. So this is the capacity to hold online the goal that is motivating behavior. So this also likely reflects recurrent networks that interact across associational regions in the frontal and the parietal lobes at least, but also the temporal lobe. Now, when we think of memory, probably what we have in mind really is long-term memory. So this is the kind of memory that reflects the accrual of life experience and its consolidation, and the capacity to then recall this information and have it once again enter our conscious deliberations. This kind of long-term memory is what is clearly dependent upon the associational networks of the temporal lobe, and specifically the medial parts of the temporal lobe. So this gets us into parts of the brain that we've just briefly encountered in our survey of the human brain in the first unit of the course. You may recall that there is a gyral structure on the medial edge of the inferior temporal lobe called the parahippocampal gyrus. This is an important gyrus for this capacity to consolidate the moment and build long-term memory. Now, deep within this gyrus, on the medial edge of the cortical mantle, is a structure that we saw in cross-section called the hippocampus. And this, as it turns out, is the critical brain structure for the acquisition of declarative memory. So when we speak of long-term memory, really what we have in mind is the storage of declarative memory and the capacity to recall those memories. So here in a phantom view of the human brain, we're reminded that deep to this parahippocampal gyrus, we have this structure called the hippocampus. And just in front of the hippocampus is the amygdala. Now, the amygdala is associated with yet other aspects of complex brain function, namely emotion. And we're going to talk about emotion in a separate tutorial. So for this tutorial, we're going to leave the amygdala out and focus in on the associational networks that are associated with this fascinating region of the mammalian brain called the hippocampus. So let's look more closely at the hippocampus. And I'll just remind you that the hippocampus is a cortical structure, but it's found at the medial edge of this parahippocampal gyrus that folds in upon itself, forming the floor and the medial wall of the lateral ventricle as it makes its shape into the temporal lobe. Now, the hippocampus is one of those wonderful terms in neuroanatomy that I'm sure many of you know. We've talked about it briefly in Unit 1. The hippocampus is a Greek term for seahorse, or perhaps sea monster, depending upon your interpretation of the Greek. One can imagine that this structure bears some resemblance to the body of a seahorse, reflecting the curving back upon itself as this medial edge of the cerebral mantle took shape in the temporal lobe during the third trimester of gestation. Now, interestingly, if you actually dissect the hippocampus out of the human brain and just lay it out on a tray, it also resembles the body of a seahorse from that view. And there are at least a few images out there that you're certainly free to Google to see that view of the hippocampus as well. Perhaps for the next version of this course, I'll do that dissection myself and be able to show you the sea monster within our temporal lobe more directly. But for this view, I would just emphasize that the hippocampus is a cortical structure, but it's the simplest of all cortical regions. Phylogenetically, we call this archicortex, which reflects its ancient origins in the evolution of the vertebrate brain. But we retain this simple architecture even in our brains. It's a simple architecture, but it's evidently sufficient to do the job of consolidating declarative memory. Now, the hippocampus receives input from this parahippocampal gyrus, and I won't go into the details of exactly how all that works. You may remember, though, that there are inputs that enter the dentate gyrus, which is this curved region where we find those dentate granule cells that continue to harbor some neural stem cells that differentiate into interneurons throughout life. So these dentate gyrus cells are being added to throughout our lives, and so there is plasticity among that cell population, both in terms of its members, but also, of course, in terms of its synaptic weights. So the dentate gyral cells then project to other neurons in the hippocampus, and there are yet additional associational connections, and it's those associational connections that we studied in Unit 2 when we looked at the synaptic basis of long-term potentiation and long-term depression. We think that these synaptic changes are the cellular and synaptic mechanisms that are responsible for consolidating memories. A memory, after all, is simply a a permanent record. So something is changed in the structure and function of circuits that enable the storage of memory. And we know that the hippocampus is an essential structure in this medial temporal lobe memory network that is essential for the acquisition and perhaps also the recall of declarative memory. So, thinking about the inputs to the hippocampus, I like to think of the hippocampus as this great funnel of the moment. Everything that defines your sense of right now, the sound of my voice, the sight of my face, the background image that you see on your device, the setting in which you find yourself in. Perhaps there are other people around you. All of these sensory stimuli define right now. And this information is being processed through the relevant sensory cortical areas. It's being fed into associational cortical regions in the occipital lobe, the temporal lobe, the parietal lobe, and even the frontal lobe. And from these associational cortices, inputs are being funneled into this parahippocampal gyrus. And from there are relays from this cortex, called the entorhinal cortex, into various divisions of this hippocampal network. And from there, through mechanisms that we think are inclusive of long-term potentiation and long-term depression, memories are stored. Now, we know much less about the actual storage of the memory than we do about its acquisition. In terms of storage, we know that the hippocampus maintains back projections through various pathways, including projections that are directed back out through the parahippocampal gyrus to each of the associational cortical regions that were relevant in the processing of the sensory stimuli that comprise the here and the now, the moment. So these extensive interconnections between associational cortex in each of the lobes and this parahippocampal gyral cortex seem to be critical for the storage and the recall of information. So when we recall a memory, and I would invite you all right now to recall some memory, perhaps a memory from childhood or perhaps a memory of what you ate for breakfast today. Whatever memory you may be operating with right now reflects the back activation from this medial temporal lobe memory system to the relevant associational cortical regions that first process that information. So there must be plasticity in the connections back from the parahippocampal gyrus cortex into these various associational regions that are associated with vision and audition and olfaction and even gustation, perhaps even the vestibular sense, although we know much less about that sense in terms of cortical processing and memory. So one can imagine the kinds of problems that patients might have with damage to this critical part of the medial temporal lobe. This produces a clinical phenomenon that we call amnesia. So amnesia refers to pathological forgetting, a failure of our medial temporal lobe memory system to consolidate new memory and perhaps even to recall memories that have previously been laid down in these vast associational networks. Amnesia can be in the anterograde direction, that is, from a moment in time of injury or disease or disorder going forward, or amnesia can be retrograde. It can project back into time prior to the moment of injury or disease and disorder. Now, for damage to the hippocampus, we will find anterograde amnesia, but also some degree of retrograde amnesia. Evidently, it takes some time for memories to be consolidated and to be laid down for long-term storage. Now, perhaps some of you, like me, have suffered a concussion at some point in your life. Perhaps some have had even more severe forms of traumatic head injury than what we might label concussion. But one common feature to traumatic injury of the brain is amnesia for the moment of impact. in the case of trauma. So in my case, I was in a motor vehicle accident last fall, and I recall the approach of the intersection at which I got into my motor vehicle accident, but I really don't recall the moment of impact, nor do I recall the approach of the vehicle that hit me from the side. I have a sense of proceeding as I had right of way into that intersection, but the next thing I remember was coming around and realizing that I'd been in an accident and now crawling out of my vehicle. So I had a bit of retrograde amnesia for the few moments leading up to the impact that resulted in my concussion. Evidently, those moments leading up to that impact were percolating in the networks of the medial temporal lobe memory system in my own head. And the trauma of the concussion was sufficient to alter the firing that was ongoing in that network. So those moments are simply lost. The activity evidently cannot be reconstructed. And as a result, I simply don't have access to those few moments of my life. Now, the concept of amnesia is one that has gained quite a lot of interest, especially among people that make movies. Some of you may be familiar with several movies that came out of Hollywood in recent years on this theme of exploring the life of someone who has suffered amnesia. Some of them are rather comical and distasteful and do a poor job representing the lives of such people, but there have been some that I think have done a realistic job portraying life with limited capacity to lay down new memories. Well, I just want to be clear. What I've been talking about thus far in terms of the medial temporal lobe memory system associated with the parahippocampal gyrus and the hippocampus itself pertains to declarative memory, the acquisition, the consolidation, and then the processing that's necessary for long-term storage of declarative episodic memories, the episodes of our lives. Well, what's been fascinating to discover about people that have had damage to the hippocampal system is that they retain memories that we might categorize as non-declarative or procedural. And that's sufficient evidence to think that there are different brain systems outside of the medial temporal lobe that mediate procedural memories. So let's turn our attention there now. So the important point that I want you to be able to understand is that when it comes to non-declarative memory, to procedural memory, these forms of memory are dependent upon entirely different brain systems than are those that are responsible for the acquisition, storage, and recall of declarative memory. So we've been talking about declarative memory and the role of the hippocampus in the short-term storage of those memories, their acquisition, their consolidation. And I mentioned that the long-term storage of declarative memory engages widespread cortical regions, many of which were involved in the initial processing of the here and the now. So when it comes to our memory systems for non-declarative memory, we're just now really beginning to do the kinds of experimental work in humans and non-human primates that would shed more light onto the kinds of systems that are essential for acquiring these kinds of memories, for consolidating them, and then for long-term storage. Now, our figure from the book indicates that, well, we don't know really what's involved with the short-term acquisition of declarative memory. I would suggest that perhaps we do know a little bit more than what this figure would imply, and I certainly want to now restore the integrity of my cerebellar hemispheres so that I can make the point that the cerebellum appears to play an important role in the acquisition and consolidation of procedural memory. One can think of procedural memory as a form of motor learning that requires the generation of error correction signals in order to acquire a memory, and that memory then takes the form of an accrued skill. at least with respect to motor function and motor behavior. And these are functions that we associate with the cerebellum, and the means by which the cerebellum can modify the operation of our motor cortical regions here in the posterior part of the frontal lobe. So I would suggest that for the short-term acquisition and storage of non-declarative procedural memories, the cerebellum is among the brain structures that will have an important role. Now, we know this because in functional magnetic resonance imaging studies, for example, when human subjects are confronted with a challenging motor skill, activity in the cerebellum is significantly modulated as these kinds of skills are being acquired. But once a certain measure of proficiency has been established, the activity of the cerebellum is much less, and that would be consistent with the idea that as we learn a new skill, the generation of error signals is quite robust. But as the skill has been consolidated, and now that skill is executed with a certain measure of proficiency, then the need for infusing additional error signals is simply less. So the cerebellum is playing an important role in the early stages of the acquisition and the consolidation of at least procedural memory. The long-term storage probably reflects, again, widespread plasticity throughout the networks that are responsible for the execution of the skill, be it a procedural skill, a motor skill, or some other aspect of implicit human behavior. So again, if we were to look into the brain anatomy that might be responsible, we'd be looking at components of the motor system. Perhaps the cerebellum continues to play an important role in the storage of skill. That might be especially true for the dentate nuclei in the lateral parts of the cerebellar hemispheres. But it would also likely engage circuitry that run through the basal ganglia and certainly the premotor cortex, among other sites. So this remains an active area of ongoing study, the acquisition of procedural memory or the acquisition of motor skill and how that changes the brain. Well, I'd like to conclude this tutorial by talking about how memory changes across the lifespan. As I've already alluded to, we're familiar with the concept of learning to ride a bicycle in childhood and never forgetting. It does appear that as long as the motor system is competent to perform motor acts, there is a indelible lifelong storage of procedural memory that seems to be relatively robust against injury and disease that would afflict the nervous system. Sadly, that doesn't seem to be the case for our declarative memory system. We know that our memory system does indeed change across the lifespan with age and is particularly vulnerable to certain forms of neurodegenerative disease. Well, to just remind you of how the brain indeed does change across the lifespan, we achieve the peak size of our human brain probably at about the end of the second decade of life. Thereafter, for both men and women, there is a gradual decline in the overall size of the brain. So what's actually changing as we age through adulthood? Well, we know from studies in animal model systems that there are at least a few cellular components of the brain that are likely responsible for this slow, gradual decline in overall brain size. So what's actually responsible for this gradual decline in the size of our brains throughout our adult years? Well, based on non-human studies, we think that one cellular element that is responsible for this decline is a reduction in the numbers of synapses that are present in our networks. As you may recall from Unit 5 of the course, our synaptic density seems to peak in our adolescent years, and throughout the remainder of adolescent life into young adulthood, there is a rather dramatic decline in synaptic density on the order of 30 to 50% in many regions of the cerebral cortex. Thereafter, the decline in synapses is not nearly so steep, but it is an ongoing process. Now, in adolescent life, the pruning of synapses, the decline in overall synaptic density, is part of normal, healthy development. It's a consolidation of networks that are responsible for who we are as unique human beings. However, throughout life, the loss of synaptic connections seems to gradually erode human performance in various dimensions of our lives, be it in motor performance or in cognitive performance, in one domain of cognition or another. Now, other changes that are taking place reflect changes in the structure of myelin around axons and white matter pathways. So while myelin is not necessarily being lost, although it may be in some regions of the cerebral white matter, myelin seems to be disrupted at an ultrastructural level. And as a consequence, the tight wrapping of myelin around axons in white matter seems to be loosened. And this in itself, we think, is an important consideration that explains functional decline across adulthood. One consequence of having this loosening, if you will, of myelination around axons is that the propagation of those signals is now subject to propagation failure, or at least to inefficiencies in the rates of transmission of information from one gray matter center to another. Now, it's also important to emphasize when we're considering normal, healthy aging that we are typically not losing neurons as our brain ages throughout adult life. This gradual decline that we see in brain size is due very likely to these kinds of ultrastructural features of networks and neuronal connections. But the actual loss of neurons, we now think, is pathological. That is to say, we normally are not losing neurons as we age. Our gray matter volumes are declining, but not because of the death of neurons. Now, sadly, there are individuals that are experiencing the death of neurons, but we consider that to be a pathological context. So cellular loss throughout the brain is not to be considered a part of normal aging. Now, I suspect that many of you may be supplementing your diet in various ways with products that are intended to increase cognitive capacity and memory function and other aspects of human brain performance throughout adulthood, or perhaps you're on particular dietary regimes that are designed to do the same. And you may be wondering, well, is there significant benefit from these kinds of dietary factors? I would suggest that we still don't understand well enough yet how these dietary factors impact brain structure and function. There may be benefit, but I think what is clear is that the benefit of aerobic fitness trumps the more modest benefits of any of these other factors that heretofore have been studied in terms of supporting brain function into adult life. So I think that's great news. I think what we need to appreciate is that sometimes the best solutions to our health problems are right in front of us and are often the most simple. And simply moving our bodies, increasing our aerobics fitness, seems to me anyway to be the most compelling single factor that will promote the health and the function of our brain throughout the lifespan. Now, quite sadly for many adults around the world, a serious concern with memory and aging is the development of dementia, and one particular leading cause of dementia, Alzheimer's disease. Now, we could easily spend an entire session in medical neuroscience together talking about Alzheimer's disease. Unfortunately, I have not planned to do so. For those of you who may have a special interest in this topic, I'll have to simply refer you to other sources that are available. Perhaps in the future we may develop a neuropathology course that will give us the time and the place to explore such diseases of the human nervous system in more depth. But for this session, I simply want to touch on this topic and perhaps inspire you to learn more on your own. Well, Alzheimer's disease is a neurodegenerative disorder that primarily afflicts the cerebral cortex. And here in this figure we see the distribution of pathological hallmarks of Alzheimer's disease, with these dots reflecting the density of those pathological changes. And what we can see is that the entire cerebral mantle can eventually be affected with Alzheimer's disease, but the disease process itself seems to be focused on the medial part of the temporal lobe and then extending into the parietal associational cortical regions. So on the lateral surface, there seems to be a broad region of the parietal temporal associational network that has severe affliction with the development and progression of Alzheimer's disease. So what these individual dots in this schematic actually represent are the two features that are hallmarks of this disorder. One is the presence of something called the senile plaque, which is an extracellular accumulation of protein and other kinds of cellular components. And in addition, the second hallmark is something that we call the neurofibrillary tangle. And this is an intracellular inclusion that is present within neurons that are very likely on their way to degeneration. And certainly when they present with a neurofibrillary tangle, that is an indication of dysfunction within the cytoskeleton of the neuron and the trafficking of important subcellular constituents that are important to support the function of the neuron. So senile plaques and neurofibrillary tangles are concentrated then in the associational regions of the medial temporal lobe and then the lateral temporal cortex into the parietal associational cortex. And the affliction of these associational regions helps us understand why in Alzheimer's disease, one of the early indications of the onset of the disorder is a failure of our medial temporal lobe memory system. The distribution of these plaques and tangles also helps us understand why long-term memories are often preserved in these patients, at least prior to the terminal stages of the disease. It's quite common to find individuals afflicted with this disorder that retain memories from childhood, but have severe disability in their capacity to acquire and to consolidate new memory. And we think this clinical picture reflects rather well on the distribution of pathology within these associational regions of the cortex. Not always, but typically we find that association. Well, again, I feel like I need to apologize to those of you that may have been drawn to medical neuroscience because of a particular interest in Alzheimer's disease or other kinds of disorders and diseases of the nervous system. It's simply beyond the scope for us to talk in depth about the various neuropathologies that afflict the human brain. I hope rather I'm giving you a solid foundation that would support your explorations of these kinds of disorders, should that be where you want your studies of the brain to go in the future. Well, we need to move on and consider one other domain of function associated with the temporal lobe, and that would be language. Again, another fascinating topic. We could have a whole course just devoted to brain and language. Well, in fact, we do. We have courses here at Duke University on this very topic. So my aim with you, rather, is to simply give you some of the basics that you need to understand functional localization in the human brain. Well, humor me for a moment, will you? And let's test your facility for the recognition of words, at least in presentation in English language form. So let's read together the following text. According to research at Cambridge University, it doesn't matter in what order the letters in a word are. The only important thing is that the first and last letter be at the right place. The rest can be a total mess, and you can still read it without a problem. This is because the human mind does not read every letter by itself, but the word as a whole. Well, for those of you who may not be native English speakers, perhaps you struggled a little bit with this challenge, but you can create your own such text in your native language and demonstrate for yourself and your friends and your family really this amazing facility of the human brain to take these arbitrary visual symbols and interpret them with meaning and semantic content, and to run them through our language circuit in a way that allows us to produce the articulated vocal sounds that are associated with these relatively arbitrary visual symbols. Well, this is evidence of the amazing facility of the human brain for manipulating symbolic representation derived from sensory stimuli and using it to communicate complex ideas and complex thought in the form of speech. So how does this work in the brain? Well, there are regions of our associational cortex, primarily in the left hemisphere, that are responsible for such amazing linguistic feats. Two regions in particular we need to discuss for just a few moments. One we find associated with the lateral part of our temporal associational cortex. And this is named after a German physician, Karl Wernicke, and most of us in the West will pronounce that as Wernicke's area. And it is often found in the superior temporal gyrus, roughly the posterior 1/3 of the superior temporal gyrus. But I think the reality is that there's not a single module in this lateral temporal cortex that we can identify as Wernicke's area. Rather, I think we need to understand that there's probably a vast network of nodal points that are critical for the recognition of these sensory stimuli and their interpretation, their encoding in terms of semantic content. So the actual critical nodes in this network may be found in any one of a number of places. And indeed, for those of you that are bilingual or multilingual, you may have distinct nodal points that represent meaning in the language that is native to your personal life history. Some of you, I'm sure, can communicate using the symbols and gestures that we call American Sign Language. And for the representation of those sensory stimuli, there may be yet another set of key nodal points that establish an associational network here in this lateral temporal cortex. So in my view, and I think this is the view of the neurosurgeons that actually map out these regions in the human brain, there's unlikely to be a single region that is consistently localized in every human brain that we can label as Wernicke's area. Rather, at least the job of the neurosurgeon will be to figure out what are those key nodal points in the networks that represent the semantic knowledge that you operate with that are essentially untouchable from a neurosurgical perspective. That is, the key nodes that the surgeon is trying to identify so that she or he can preserve those nodal points during the course of an operation. So from this point, I think this figure from our book is just a little bit misleading. I would suggest that there's not a single module that we can localize and call Wernicke's area. Rather, I believe that there is a more extended network in the associational cortex of the lateral temporal lobe that is processing these sensory symbols that are derived primarily through vision and auditory senses, and then interpreting them and applying semantic meaning to those sets of symbols. And this network then does the job of encoding the meaning of these symbols. And in terms of language function, this is where the understanding or the comprehension of language is built up, in the human brain. So I think you can understand what might happen then if there would be some damage to key nodes in this lateral temporal associational network. One might still have the capacity to make speech, but one would expect severe impairments in the ability to comprehend speech. Now, our focus in this session is indeed on the temporal associational cortex, but I can't help but to jump across the lateral fissure and get into an inferior lateral region that we call Broca's area. Broca's area, as many of you know, is an area that is involved in the production of speech. Broca's area was named after Pierre-Paul Broca, who described a patient with damage to this part of the brain. And in life, following this injury, the patient was severely impaired in his capacity to make speech. Now, we now understand Broca's area really as part of our premotor cortical network. You'll recall we described a mosaic of frontal cortical areas that are associated with the planning of movement, especially movement that's directed away from personal space. So this aspect of planning for movement also applies to the governance of our vocal articulatory apparatus in our throat, our larynx, our pharynx, the coordination of our breathing muscles. So this is, in a sense, a premotor cortex that interacts closely with the nearby regions of the precentral gyrus that have upper motor neuronal control over our vocal motor apparatus. So from that standpoint, we can understand Broca's area as a premotor cortex for vocal articulation. But it's not just for vocal articulation. Broca's area also seems to be involved with the production of written language, which would have it affiliating with not just the lateral segment of the precentral gyrus, but also the middle segment of the precentral gyrus that governs the movements of the distal upper extremities, the part of our body that we use to write with. So damage to this part of the inferior frontal lobe, specifically what we're referring to is the posterior 1/3 of the inferior frontal gyrus. And there are various subdivisions of this part of the inferior frontal gyrus that we could identify. I think from neurosurgical procedures, it seems clear that there may be a set of nodal points within the networks of this posterior inferior frontal gyrus that are critical for the expression of language. So again, like Wernicke's area is not a single module that we can point to and identify, I think the same is likely to be said of Broca's area, although the localization seems to be much more consistent from one person to the next in our brains than would be Wernicke's area in the lateral temporal cortex. So here's roughly the location of Broca's area in the human brain, and this is a region that, if damaged, can produce a severe impairment in the fluency of speech. With an understanding then that Broca's area can be conceived of as an essential premotor network for the production of speech, one can imagine that damage to Broca's area would lead to a severe inability to produce speech, not just speech in oral form, but also the written language will also be affected with damage to Broca's area. So it's critical for anyone headed towards a career in the health professions, or indeed practicing in the health professions, to be able to differentiate between two forms of impairment that are associated with these critical language areas in the human brain. So an impairment of language function is called aphasia, and if the impairment is afflicting the lateral temporal cortex, then we call this Wernicke's aphasia. If the injury is to the inferior frontal gyrus, then the aphasia that we would expect is called Broca's aphasia. So let's differentiate these two conditions. So the key differentiation between these two forms of aphasia is that with injury to the inferior frontal gyrus in Broca's aphasia, comprehension is intact, but there is a severe impairment in the fluency of speech. Whereas with Wernicke's aphasia, the situation is somewhat reversed. There is fluency of speech, but the capacity for comprehension is severely impaired. Now, these two parts of the brain, the lateral temporal associational cortex and the inferior frontal gyrus, are interconnected by white matter structures that span this space within the brain between this temporal lobe around the lateral fissure of the brain and into the posterior part of the inferior frontal lobe. One can have damage of those white matter pathways and see some combination of these deficits that reflect the failure of communication between this lateral temporal associational cortex and the inferior frontal gyrus. One might also have, let's say, a stroke involving the entire distribution of the middle cerebral artery and have a global aphasia that affects both the lateral temporal associational cortex and the inferior frontal gyrus. So while it's possible to have one type of aphasia or the other, one can see elements of both, or one can see a global aphasia impacting all aspects of language function. Now, since we're talking about aphasia and Broca's aphasia in particular, I just can't help but to point you to a fantastic video that was juried and awarded the grand prize in our inaugural Brain Awareness Week video contest, sponsored by the Society for Neuroscience. And this is a beautifully produced and just wonderfully rich story told by a graduate student in the field of neuroscience from Australia. Her name is Cherie Heath, and she's telling the story of her grandfather's stroke, who suffered from a Broca's aphasia. So I would encourage you to just take a few minutes, navigate to this page at brainfacts.org, and you can search aphasia or the treasure hunt, which is actually the title of this short video. And I think you'll agree with the judges of this contest that Cherie was very much deserving of the grand prize, given her insight, given the accuracy of the neuroscience in her presentation, and of course her marvelous creativity with which she pulls all this together. And in the end, you'll get a chance to explore and reinforce some of what we've been talking about here recently about the language centers of the human brain. Now, as we've been discussing these important structures for the production and the understanding of human language, I've been highlighting the left hemisphere of the brain. And one final point that I'd like to leave with you is the lateralization of language function in the human brain. And this gets us into a broader discussion of cerebral dominance that is, again, unfortunately beyond the scope of medical neuroscience, but perhaps you'll be inspired to learn more about that topic on your own. But at least for language function, it's important to emphasize that those functions that we associate with Wernicke's area and Broca's area are lateralized to the left hemisphere for the vast majority of us. For those of you that are right-handers, as am I, almost certainly your functional Broca's and Wernicke's areas are present in the left hemisphere. For those of you that are left-handed, then the probability of your Broca's and Wernicke's area being present in the left hemisphere is more like 80%. So really, for the vast majority of people on the planet, these functional regions are present in the left hemisphere. But of course, what you really care about is what is the situation in your own brain. Unfortunately, most of us don't get an opportunity to really confirm the organization of our language areas unless we've had a brain injury or unless we've been subject to a study that would reveal the organization of these parts of the brain in the two hemispheres. So for the right-handers out there, you can be quite confident that your functional Broca's and Wernicke's area are lateralized to your left hemisphere. Now the situation with left-handers is a bit different. This remains an active area of investigation. It's not that left-handers have a reversed pattern of cerebral lateralization. It's that the lateralization seems to be less severe or perhaps less complete. So again, about 80% of left-handers have their language centers present in the left hemisphere. So you should be wondering, well, okay, if you're like most of us and you have Broca's and Wernicke's areas in your left hemisphere, what about the right hemisphere? Are there corresponding brain regions? And if so, what's their function? Well, indeed, there are corresponding brain regions that are present in the right hemisphere that occupy the same position in the lateral temporal associational cortex in the case of Wernicke's area, or the inferior frontal gyrus in the case of Broca's area, but there may be differences in the organization of these networks. One very striking difference that's present in most of our brains concerns the superior surface of the posterior temporal lobe, a region that we call the temporal plane, or in Latin, the planum temporale. So this posterior temporal plane, which is highlighted here in blue, is larger in the left hemisphere compared to the right hemisphere for most human beings. Now, a simple interpretation of this is perhaps this asymmetry in planum temporale size is what instantiates our Wernicke's area in the left temporal associational cortex. Well, for a variety of reasons, we think it's not quite that simple. In fact, the asymmetry may be important for serving other aspects of auditory function. One intriguing finding, now about a decade old, is the idea that for those individuals that have this amazing capacity for perfect pitch perception, that their asymmetry is especially pronounced. So these would include certainly trained musicians, but not just skilled musicians. There are other people out there who have this capacity, and there are certainly wonderful world-class musicians that lack this ability. But nevertheless, there seems to be something important about planum temporale asymmetry that is one means perhaps of consolidating more circuitry for efficient processing in one hemisphere, rather than more broadly distributing it across the hemispheres and thereby mandating communication through the corpus callosum. Well, so getting back to the functions then of the right hemisphere, we do in fact have corresponding temporal associational cortical regions and an inferior frontal gyrus in the right hemisphere. And for those of us that are strongly lateralized to the left, our right hemisphere does contribute in important ways to language function, just not in perhaps the most overt means that is accessible to simple clinical testing, such as comprehension and speech production. Rather, what we believe is that this right hemisphere is especially involved in the emotional aspects of vocal communication. We believe that the temporal associational cortex of the right hemisphere is important for interpreting the prosodic quality of speech, that is, the emotional content of speech. And likewise, in the right-sided language regions of the inferior frontal lobe, we have networks that seem to be responsible for imbuing speech with emotion. So the right hemisphere seems to be very important for that quality of communication that relies more on emotion than it does on the semantic lexicon that we access as we produce speech. And lastly, one additional component of lateralization of function that's worth mentioning is that there seems to be gender difference in the lateralization of language capacity in the two hemispheres. Women tend to be somewhat less severely lateralized compared to men. One reason why we believe this to be the case is that following a left middle cerebral artery infarction, the capacity of the female brain to recover language function seems to be greater than it is in the male brain, suggesting that the lateralization of language function in the female brain might be less complete than it is in the typical male brain. Now, some have taken this idea to the extreme and have suggested that women simply are more flexible in their ability to mobilize resources that are distributed across the corpus callosum in the two hemispheres. I'm not so certain that we can generalize to that point yet, but it does seem to be an intriguing possibility that with lateralization being less complete in the typical female brain, perhaps that confers some advantage in accessing cortical circuitry via callosal connections that might mobilize additional resource to solve complex problems and to manage everyday life. So, you women out there, you may take some comfort in knowing that perhaps you have more ready access to the resources in your two hemispheres. And for the men out there, well, some might say that we are stuck in one hemisphere, and given the rate of loss of callosal fibers, we might imagine that our hemispheres are slowly drifting apart, as was the point of a cartoon I saw once. Well, I think that's going a little bit too far here, but it is worth considering the degree of lateralization in the human brain, and there are other dimensions of lateralized behavior we've talked about to some extent. We talked early on about human-handedness and how that might be reflected in the structure of the sensory and motor cortical regions. We've talked recently about the lateralization of attentional capacity to the right hemisphere. That would help us understand why the hemi-neglect syndrome is most commonly seen with right parietal damage, affecting our ability to interact with all things left. And now we add language to the story of lateralization, recognizing that for most of us our language function is rather severely lateralized to the left hemisphere, but there are still complementary functions of the right hemisphere that are necessary to have a well-rounded, integrated capacity to communicate effectively, not just with the semantic content of our speech, but also with the emotions that are so critical for conveying genuine human-to-human interaction. Well, there's so much more we could have said about human memory, about brain and language. I know we've only scratched the surface, and I would encourage you, if that is an area of special interest to you, to pursue those studies through other resources, other means. Hopefully we'll have other kinds of online courses available that will allow you to pursue your specialized interests in these dimensions of cognitive neuroscience. But for our purposes here in medical neuroscience, I want to focus on the anatomical framework that I've tried to provide for you. So it's critical for you to understand the localization of the hippocampus and the parahippocampal gyrus and the function of that brain network in acquiring and consolidating declarative memory. Likewise, it's critical that you be able to recognize the major language centers in the human brain and the types of aphasias that result with localized focal injury to the lateral temporal associational cortex and to the inferior frontal gyrus. So, with those localizing points of emphasis in mind, I think we can wrap up now our session on the temporal associational cortex and anticipate our next time together, where we will indeed continue to explore the associational cortex of the frontal lobe and consider yet additional dimensions of brain function that pertain to cognition. So I'll see you next time across the lateral fissure into the anterior part of the associational network. Welcome again to Unit 6 of Medical Neuroscience, where we have been discussing the associational cortex of the cerebral hemispheres. And in this session, we turn our attention to the frontal lobe. And I'd like to speak with you about the associational cortex that we find in this very large and very important part of the human brain. Our topic today pertains to the two core concepts in the field that have been especially salient for Unit 6 on complex brain functions. It's through the integrated action of the associational cortex that intelligence arises in the brain, especially in our brain, where our associational cortices are so well formed and well evolved. And this intelligence allows our brain to reason, to plan, and to solve complex problems. That's not to say that other brains aren't capable of that. Indeed, in insect colonies, even with their ganglia, it's hard to call what they have a brain, but one may choose to do so. But even in the central nervous system of insects, we see some capacity for each of these dimensions of cognition. But certainly for us, much of what we associate with the intelligence of our species is attributable to the integrated action of our associational cortices. And it's our human brain that endows us with the curiosity that drives us to explore our world and even pursue studies of things such as medical neuroscience. My learning objective for you in this tutorial is to discuss the major functions that are localized to the associational cortex in the frontal lobe. So in this tutorial, we're going to turn our attention to the frontal lobe. So I'll just remind you that the frontal lobe is bounded posteriorly by the central sulcus. And here we see the central sulcus continuing into the medial face of the hemisphere and the paracentral lobule. The frontal lobe is bounded inferiorly by the lateral fissure, which is also known as the Sylvian fissure, separating the frontal lobe from the temporal lobe. Now, when we look at the frontal lobe, we realize that there are really two major cortical territories that we find in the frontal lobe. Here would be our central sulcus, dividing the frontal lobe from the parietal lobe. And as you know, at the posterior aspect of the frontal lobe, we have our motor cortex. So anatomically, that's localized to the precentral gyri, and then to the posterior part of the superior, middle, and inferior frontal gyri that harbor the premotor mosaic in the two hemispheres. So the posterior part of the frontal lobe is essentially our motor cortex. So when we talk about the associational cortex of the frontal lobe, basically what we're describing is all of this cortex that sits anterior to the motor cortex. And that would, of course, include the cortex that we find on the medial face of the hemisphere. Now, we do have an unusual term that we use to talk about this part of the frontal associational network. We call this the prefrontal cortex. So you may be wondering, well, that's an odd word. Prefrontal, what does that mean? Well, prefrontal essentially means cortex that is anterior or before the motor cortex. So for our purposes, the prefrontal cortex can be localized anatomically as the anterior two-thirds of the superior, middle, and inferior frontal gyri. Remember our three fingers here on the dorsolateral aspect of the cerebral cortex. So roughly where my three fingers are, that would be the prefrontal cortex as seen from this dorsolateral view. And then from the medial view, the prefrontal cortex would include the cortex of the superior frontal gyrus and the cingulate gyrus as they wrap around into the medial part of the orbital cortex of the brain. including this gyrus rectus here right along the midline on the ventral aspect of the frontal lobe, and including an interesting little region right here called the subcallosal area, right underneath the genu of the corpus callosum. Now, the posterior boundary of this prefrontal cortex would be, again, roughly about 2/3 of the way along the length of the cingulate and superior frontal gyri. We would recognize the premotor cortex sitting just a few centimeters in front of our paracentral lobule. So once we get roughly anterior to about this location, we're dealing with our prefrontal cortex now as we see it on the medial face of the hemisphere. Now, we also, of course, include the orbital surface of the brain, and our orbital cortex is part of our prefrontal associational network of the frontal lobe, all the way back to where the cortex of the frontal lobe gives way to the basal forebrain region and the hypothalamus. Now, for our purposes, I'd like to further subdivide the prefrontal cortex into two major sectors. So the dorsolateral prefrontal cortex is where our executive functions are governed. And by executive functions, what we really mean are those functions that are associated with planning behavior based on the integration of sensory signals with other sources of information that would include our memory, our emotion, our interoceptive signals. And the integration of these signals gives rise to plans for motor action. These functions are exemplified in the phenomenon that we call working memory. You'll recall that we can think about the temporal dimension of memory and realize that there is a period of time from maybe seconds to minutes where it's possible to maintain in consciousness some goal that can motivate our behavior. So this is what we have in mind by working memory. Imagine, for example, that you're looking for a lost object. Perhaps it's a set of keys, or perhaps it is a coin or some other possession that is of value to you that motivates your desire to seek it out. That process of search, wherever you may be, in your home, in a yard, in a public place, that search is being motivated by the maintenance in your consciousness of that object, that goal that's motivating your behavior. So this is the concept of working memory. Yes, it's dependent on your medial temporal lobe memory system, but really what's going on is this object is now being maintained in awareness in the circuitry of your dorsolateral prefrontal cortex so that it can guide the appropriate search behavior. Well, we've studied this in various ways in animal subjects and in human patients, and I'd like to share with you some really seminal work done by Patricia Goldman-Rakish and her colleagues at Yale University in the late 80s and into the 90s. This work really began to develop a neurophysiological model for working memory. And what Dr. Goldman-Rakish and her colleagues did was to record from the cortex that is found in the banks of the principal sulcus in the rhesus monkey brain. So this would be roughly equivalent to recording from the inferior aspect of the superior frontal gyrus in the human brain. And what they were able to demonstrate with a very intriguing behavioral paradigm is that there are neurons whose firing activities seem to represent the goal of the behavior that's held in working memory. So here's the paradigm. Animal model, a rhesus monkey, is observing the placement of a morsel of food in one of these two wells that are within arm's reach. So the monkey's aware of in which well the food was placed. So now a delay is interposed between the observation, the sensory information coming into the animal, and the ability to execute the appropriate motor behavior, in this case a visually guided reach to retrieve the food reward. So this delay is imposed by a screen that's lowered for some period of time, typically some number of seconds. So after some period of delay, the screen is now raised, and this monkey is now free to uncover the well of choice and retrieve the food morsel. So there's this period of time, this delay period, where the working memory of the animal is exercised. The goal of the pending behavior is held online until that time when that behavior can be executed, in this case a visually guided reach. Well, what Dr. Rakesh was able to demonstrate is that there are neurons in this dorsolateral prefrontal associational network whose firing rates are modulated by this delay. So here's recordings from a single neuron in this part of the brain, and what we see is that the firing rate is elevated during this time of delay. So the delay is indicated by this region here that is shaded red in the background. And so when there is a food morsel that is loaded into the well, and the monkey can see that, we see that the activity of this neuron rises during the delay period. And immediately following the delay, when the behavior is to be executed, there is a sharp fall off in activity for this cell, suggesting that what this neuron is encoding is the information that is going to guide the execution of the behavior when it's possible to do so. In other trials, when the food was not introduced, simply lowering the screen and after a delay raising the screen didn't really seem to modulate this neuron's firing much at all. So this neuron doesn't necessarily seem to be responsive to the sensory elements of the stimulus. It's not necessarily encoding the dynamics of the movement per se. What it's doing is it's holding online information that is motivating the behavior. Well, why should this be important? Well, I think that much of our daily lives are actually guided by working memory tasks, such as this one. Well, we may not be often retrieving a food reward after an imposed delay, but we're constantly planning for the future. And those plans, be they short-term or long-term, are predicated upon being able to represent the goal that's motivating our actions. Well, the prefrontal cortex is very much involved in organizing behavior for future plans, both short and long term. And one should not be surprised then that this is a part of the brain that's relatively slow to develop. In fact, the prefrontal associational cortex is among the last portions of the brain to really fully achieve adult volumes for both gray matter and white matter, and also the adult degree of myelination that gives our white matter pathways their structural integrity and presumably therefore the efficiency by which signals can propagate and that information can be processed. Now, once this dorsolateral associational network is fully mature, it plays an important role in making use of prior experience, prior information, to guide subsequent behavior. So working memory is one example of this, but there are also other real-world examples that are worth some consideration. So imagine a developing child going through adolescent years and learning about the rules that govern our social relations, our culture, our society. And certain behaviors are usually considered to be appropriate and acceptable, and others are not. And this developing child, adolescent, is learning to integrate those rules and to moderate behavior, such that appropriate behaviors are shaped by these social norms that we acquire typically in the first couple of decades of life. So this dorsolateral prefrontal cortex, as it turns out, also has a very important role in moderating or suppressing behaviors that would otherwise be considered to be inappropriate. I think more generally speaking, we can recognize the importance of this capacity to help us navigate our complex, dynamic lives in the real world, where sometimes the contingencies for reward and punishment might be changing. So here's a laboratory test that is aimed at sort of getting at some of these dynamics of sort of understanding the rules for successful behavior and being able to respond appropriately. This challenge may be familiar to some of you who've taken psychology courses, perhaps even experienced this test yourself. This is called the Wisconsin card sorting task. And what's going on here is that a subject has to sort a deck of cards by placing these cards into one of a few different piles based on some kind of rule, some kind of sorting rule that is never made explicit to the subject. Only by feedback on each trial is this subject informed of the rules by which sorting must occur. So one can imagine with a set of cards such as this, one can sort by the color of the card, putting all the red cards in the same stack, all the green cards in a different stack, and so on. One can imagine sorting by shape, where all of the cards that bear star shapes are put together. All the cards that bear triangles are put into the same pile. Or one might imagine sorting by number, with all the cards that have one object on them put together, and all the cards that have two are put in a separate pile, and so on. Now, the key to this experiment is that the rules for sorting the cards are determined by the person running the experiment, and the subject is only provided feedback about whether the sorting of the card was correct or incorrect according to whatever rule is in effect. So the way this might go, for example, is that the experimenter may decide that the sorting rule is going to be by color. And so the subject begins to sort, and the experimenter may say correct, correct, incorrect, 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 and with some number of negative feedback prompts, then the subject might realize that maybe the sorting rule has just changed from sort by color to perhaps some other scheme, such as sort by shape. And by giving this feedback following each placement of a card, the subject accrues the knowledge that will guide future behavior until there is negative feedback again that prompts a change. So you can imagine what would happen with patients that have problems with their dorsolateral prefrontal cortex. They tend not to be able to integrate these changing sensory signals that would guide now appropriate behavior. They have difficulty integrating these signals to guide appropriate behavior in the future. As a consequence, these individuals tend to perseverate, meaning they tend to repeat the same behavior even when the rules that would shift behavior have changed. So if a person who had damage to this dorsolateral sector of the prefrontal cortex was tested in this Wisconsin card sorting task, we may find perseverative behavior. They may continue to sort according to a particular rule that may seem to be working and reinforced, even when those rules for reward and punishment may change. So these subjects have great difficulty suppressing what is now an inappropriate behavior because of a failure to properly integrate the cues that are helping to establish an appreciation for the rules that ought to moderate our behavioral choice. Well, I want to take you through a few slides that illustrate in a simple way the functions of this dorsolateral prefrontal cortex, and it gives us a chance to maybe feel a little of the dissonance in ourselves that is an indication that this network is actually working correctly. Now, I'm going to use color here, so I apologize to those of you that have some deficit in color vision. I hope you can still follow along and appreciate as best you can. But here's what I want you to do. I want you to simply report the dominant color that you see on the next sequence of slides. And go ahead and say it out loud. I'll say it with you, okay? So here we go. red, green, blue, red, green, blue, green, blue, red. Now, I've been practicing this for the last couple of minutes, and you might have experienced what I just felt, which is just a little bit of dissonance as we read a certain bit of text and our vocalization is not what we have read, but rather the color of the type that we see before us. So it's a bit of a challenge for me to look at this red-lettered blue and report the color without simply reading the word. So this is what our dorsolateral prefrontal cortical network is doing for us. It's helping to maintain in working memory the rule that's motivating behavior. In this case, the rule was to report the dominant color. There are a variety of other behavioral options that are before us at all times. And sometimes it's very tempting to deviate from the appropriate behavior and let loose what would be a behavior that perfectly legitimate, but it would violate the rule under which we are trying to operate here. So people with damage to this part of their prefrontal cortex would have great difficulty suppressing the dominant cues that would guide their behavior. And in this simple version of a dorsolateral prefrontal test called the Stroop test, often the dominant signals come in the form of the written word, the text. So it would be very difficult for a person with damage to this part of the brain to read the word blue in red type and do anything but report that word blue. So now I think we're ready to leave this dorsolateral sector of the prefrontal cortex and spend just a few minutes introducing the orbital and the medial aspects of the prefrontal cortex. Now, these parts of the associational network of the frontal lobe bear many functional similarities to what we've just been describing for the dorsolateral sector, but there seems to be some very important differences that pertain to the inputs and outputs of these cortical networks relative to the sources of the information and the effector systems that they gain access to. The dorsolateral prefrontal cortex is working on explicit information. That is information derived from our special sensory systems, from our memories, from other aspects of cognition that we can access with our language systems. We can talk about them, we can give them names, we can describe them with our verbal faculties. The orbital and medial sector of the prefrontal cortex, however, seems to be operating principally on implicit information. Information derived from our emotions that are informed by our visceral senses. Information that is united through these prefrontal networks with our faculties that are involved in planning for the future, but those plans are being motivated by these sources of information that are just more difficult to describe. Consider, for example, the challenge of articulating in verbal terms the content of an emotion, such as anger or fear or happiness or sorrow. I can do a much better job describing, for example, the texture, the dimension, the color of this object that I'm holding in my hand than I can use my verbal faculties to reveal for you the content of the twinge of anxiety that I may be feeling at this very moment. So our orbital medial prefrontal cortex is engaged in the processing of emotion and integrating that emotion with our faculty for reason, for decision making, for planning for the future. Both our short term as well as our long term. And it's this future thinking aspect of associational processing in the prefrontal cortex that can lead to advantageous decisions as our lives unfold, or perhaps disadvantageous decisions if our motivations are simply driven towards short-term gains, impulsive action. So part of what this orbital medial prefrontal cortex does is suppress the impulses that might lead to the acquisition of short-term gain at the cost of a more long-term strategy that would lead to advantage, be it in the domain of social relations, in financial matters, or whatever the dimension of life that we might want to consider. We're going to have much more to say about this orbital medial sector of the prefrontal cortex when we talk about emotion. And we'll consider how the amygdala in the anterior part of the medial temporal lobe has an important role to play in the experience and expression of emotion, and how the amygdala is intimately connected to this orbital sector of the prefrontal cortex. Now, a modern understanding of the importance of this orbital and medial part of the prefrontal cortex and integrating emotional experience with our decision-making faculty began with the story of one individual who passed away about 150 years ago. His name was Phineas Gage, and I'm sure he's known to many of you. Phineas Gage was an individual who suffered a horrific injury to the front part of his cranium, involving facial structures and, amazingly, a huge penetrating injury to the cranial vault that resulted in damage to the orbital and medial sectors of the prefrontal cortex. Now, this injury was reported in the local newspaper in Rutland, Vermont. That paper was called the Vermont Mercury, and one Friday edition in 1848, October 13th, led with the headline, "A Wonderful Journey Through Skull and Brains." Well, much has been written about Phineas Gage, especially in the last decade, where we recognize Mr. Gage as really the pioneer of a new cognitive science that has led to a deeper understanding of the brain mechanisms that integrate emotional experience into our decision-making faculty. And arguably, contrary to major streams of thought, at least in Western philosophy, emotion and reason really need not be at war with one another, but rather in the integrated actions of our associational networks of the prefrontal cortex, we see that emotion and reason really work hand in hand to guide our decision-making in real-life situations, especially when there's some risk involved, when there's something at stake. The idea that we're going to elaborate in a subsequent tutorial is that emotions play a critical role in biasing our decision-making faculties. And I think there's mounting evidence to support this view. Still quite controversial, but nevertheless, I think it's giving us a much more integrated sense of how emotion and reason work hand in hand to moderate behavior. So I look forward to having that discussion with you as we explore the emotions and the brain mechanisms that come around the experience and the expression of emotional behavior. So until that time, I hope you have an interesting moment reflecting on the study question that I'll leave you with. And perhaps it might be necessary for you to review just a little bit of anatomy, especially with respect to the dura mater. That's a bit of a hint. So I'll see you next time as we pick up the story of complex functions of the human brain.